And then once the sucrose molecule already enter into the cis-tube element, the sucrose molecule will start to transport in the phloem cis-tube element into the sink. So any part lah, like shoot, like root or fruit, all that the cells that require the organic nutrient for metabolism, or the cell that can store the energy, store the nutrient. Okay, so once the already enter the cis-tube element, translocation will be occur. So normally one of the way of the translocation is pressure flow hypothesis. So what is this? So if you see this diagram, this left hand side, so the first step is the step that I already mentioned this at here. So it is the sucrose molecule when your sucrose molecule is already transported into the cis-tube element. Okay, so your first step is to that your sucrose will be actively loaded into the cis-tube element. So this process is you are using required ATP lah, because it is low concentration gradient into high concentration gradient. So this is why that it using the ATP, because it actively transport. Okay, so when they are already when the cis-tube element sucrose molecule is already enter into the cis-tube, next your sucrose molecule need to be transport into the sink, right? So how can be transport? How it will be transport? So it require the water molecule to help the sucrose molecule able to be transport. You need to know that sucrose molecule is organic nutrient. It is not a solution. You need to know that sucrose is not a solution. It is like a sugar. So if you imagine that if no solution, no water molecule or any liquid flow molecule, sucrose molecule cannot simply directly go from the leaf into the root one. Cannot. So it require a movement. It require transport lah, like it require a car or any transport car to transport the sucrose molecule from the leaf into the sink. Okay, so this one you will require water molecule. So the water molecule out from the xylem vessel. So normally xylem and phloem is located side by side, but the transport process is opposite direction, because we know that xylem transport water from root to shoot, and phloem transport nutrient from leaf to root. Okay, so it is totally opposite direction. So when the water molecule is already when the water molecule in the xylem vessel now is high, we know that water molecule in xylem vessel is high compared to phloem, right? This one is everyone know in logically, because xylem vessel only containing water, so automatically your water molecule in the xylem vessel will be high water potential compared to phloem. What? So this one will cause that the water molecule will diffuse out from xylem vessel and flow into the phloem cis-tube. Okay, so this one will increase your pressure force, the pressure to force the fluid pass through from the leaf into the root. Because now your when the water molecule is already come into your cis-tube, meaning that your water potential value now is very high, and then we know that water is moved down the concentration gradient by osmosis process, so that the water molecule now will be transport the sucrose molecule down to the sink. Okay, when the sucrose molecule is already reached the sink, your sucrose molecule will actively transport or passively transport unloaded into the sink. So normally any cell that require the organic nutrient, it will have this unloading process. So it can be active transport or passive transport. So it need to depending what kind of cell that the unloading process is occur. But for your syllabus, you don't need to know lah which type of cell is actively transport, which type of cell is passively transport. You just want to know that both process, both transport can be occur inside the sink for unloading process. Okay, so when your sucrose molecule now is already unloaded into the sink, meaning that your water potential value now will be very high now, because all the sucrose, your solute potential now is decreased now, and your water potential will be increased, so that your water molecule from inside the sink for the phloem sieve tube will be diffused back into the xylem vessel again. So this is one type of recycle process lah. So once water is in the xylem, transpiration pull the water to leave again. So you can say that it is one type of recycle process based on the concentration gradient lah. So normally, as I repeat again, when sucrose molecule is already pumped into the phloem molecule, the solute potential now is very high, so that the water molecule from the xylem vessel will be diffused out from xylem vessel and then flow into the phloem tube. And when there are many water molecule already accumulated here, high water potential will force the fluid pass through the sieve plate and then transport the sucrose from the sources to the sink. So when they already enter the sink, the sucrose molecule will be transported into the root cell or any type of the plant cell that require the organic nutrient. And when the sucrose molecule is already entering the sink, so that the solute potential inside the phloem sieve tube now is very low. Is already very low. So conversely, your water potential will be very high. So the water potential will not accumulate here. The water potential will be diffused out from sieve tube element and then diffused back into the xylem vessel, and then this process will be recycled again and again. Okay, so this is the general concept of the pressure flow hypothesis. So normally this is the explanation. Okay, so normally in cold transport involve phloem loading, your sugar will be from a low concentration to high concentration region, right? So normally your sugar will accumulate in the sieve tube element, resulting lower water potential. Yeah, why? Because your sucrose is already accumulated at here. So that when your sucrose molecule is already too accumulated at here, solute potential will increase, right? And then, as I mentioned before, sucrose is not a fluid; it is the sugar, so that it cannot simply transported from the phloem to the sink by facilitated diffusion if there are no fluid at there. So it requires water. So water comes from where? From xylem vessel. So why water from xylem vessel can be diffused from xylem vessel into the phloem sieve tube? Because the water potential inside the sieve tube near the source is very low now. So that the water potential inside the xylem vessel will be transported from the xylem vessel into the phloem sieve tube. So that your hydrostatic pressure now in the sieve tube near the sources is very high, so it will force the phloem sap, your organic nutrient and your sucrose molecule flow along the sieve tube to the sink. So normally at the sink, your sugar will be unloaded by both active and passive transport from the sieve tube element. So when your sugar is lost, your water potential will be increased, so that your water will be diffused back into the xylem vessel again. Okay, so this is overall of the pressure flow hypothesis. So normally another name of this theory is we call as mass flow hypothesis. So both of them actually is same, just that mass flow hypothesis is based on the experiment. Pressure flow hypothesis is we already applied inside the plant. But mass flow hypothesis, we will more focus on the experiment based explanation. So normally this one is very famous when it coming out in objective question lah. So normally objective question, it will come out like this, with this diagram, and then it will need you to give you many statements lah. It will give you many statements, and then you need to identify which statement is true or false based on the mass flow hypothesis, or known as bachelor flow hypothesis. So normally this one concept is similar, but what I say just that it is using the experimental setup to explain the hypothesis. Okay, so normally you see here there are A and B, right? So this is what we call as the permeable membrane, A and B, and then there are the beaker, right? Will contain the beaker containing water. So you need to know that what is A, what is B, and then this one is one of the examples. So maybe it will have a reverse direction one. So the most important is you need to see an arrow lah during your exam. So normally in exam they will give you a arrow, or they will color it. So normally if you see the color, the darker color will meaning that it is more concentrated lah. The lower color one, the light color one, will be less concentrated, more dilute. So if you see this diagram, you know that the water part conform is xylem vessel, because we already know that only the xylem vessel containing water. Phloem tissue does not containing water one. Not say that does not containing water, does not containing much water like this. Only the xylem vessel containing water much like this. So we know that outside the surface that this all water molecule, we assume that it is xylem vessel. And then this A is concentrated. You see the darker color, and you see the arrow here. This arrow meaning that the water is diffused into the A, into the A. So what does it mean? It means that A is the source. So in plant, if you apply into the plant, meaning that it is the leaf lah. So why it is a leaf? Because water molecule is diffused into the A. So why water molecule diffused into the A? Because your A now is very highly concentrated. So highly concentrated meaning that there are many organic nutrients inside A. So when there are many organic nutrients inside A, meaning the water potential inside the A is very low. So that the water molecule from the outside this beaker, from the outside beaker, will be diffused into flow into A by osmosis process through this permeable membrane. And then once the water potential is already increased, so that it will transport the nutrient. You see that it will transport the nutrient. You see this arrow. The solution will be flow from A into B. Okay, so when the solution is flow from A into B, so this one is what structure? Phloem sieve tube element, right? So this is the phloem sieve tube element structure, and then this flowing is flow of solution, meaning that our phloem sub is transported from the source to the sink. So when the phloem sub solution organic nutrient already reach the sink, what happened is you see that the organic nutrient now is very low. Because at the journey of the process, your organic nutrient is already diffused out diffused out and become dilute. So when they are become dilute, meaning that your water potential for B is very high. Now, so that the water molecule will be diffused out from B to outside of the cell lah. Outside of the cell. So normally is that the water molecule will diffuse back into the xylem vessel. And then you see this diagram again, so that the water molecule will be transported again from the sink, from the root into the leaf again. So this is the general experimental concept for the mass flow hypothesis. So for this kind of question in examination, normally the first important point is you need to see the arrow first. You need to determine which part is source, which part is sink. So for this one, A is source, B is sink. So it can be that, it can be the balik one, it can be reverse one. Not necessarily A is left hand side. It can be A at right hand side one. So based on the question lah. So you need, the most important is you can see the arrow, and another one is you can see the color. So the darker color is the source, the lighter color is the sink. So and another one is the flow of the direction inside the water also. So the flow or direction should be, water should be flow from the sink to the source. So you see this kind of direction, you know that B is sink, A is source.